Pleasure to be here. Good morning. I'm going to spend some time talking about our vaccine, our antibody program for HIV, passive antibody therapy. And at the end, if we have some time, if it's okay, if he doesn't stop me, I'll say a few words about what we're doing with Zika virus. The VRC is composed of several different components. The basic research building, Building 40 on the NIH campus, it houses the work of the principal investigators. But we also, just north of Bethesda, have a process development lab which helps us take our concepts for vaccines and antibody development into a manufacturing mode. And then there's a pilot plant for GMP manufacturing just north of there. And so that allows us to take our concepts into manufacturing and into the clinic, which is housed in the clinical center building. Here's Building 40, just as a comparison. And then there's another lab that does GLP assessment of our clinical samples. And this cycle allows us to work on many different vaccines. We were founded primarily to develop an HIV vaccine, which we have not done yet, but which I'll say a few words about in terms of progress. We've worked on a number of platforms, primarily gene-based approaches for these types of pathogens, and then virus-like particles for chikungunya and other alpha viruses. It's now in phase 2 testing in the Caribbean. We've been working on monoclonal antibodies, and I'll tell you today about the program for HIV monoclonal antibodies, but we also have a monoclonal antibody for Ebola now, and we're working on those for MERS and influenza. We're now starting to make protein-based vaccines, including on nanoparticles for influenza, either full-length HA or stabilized stem HA for universal flu vaccine concepts, and a subunit protein for respiratory syncytial virus. So there's many ways of preventing HIV, and there's been a lot of progress, and I'm showing you that here, all the different things that have worked to prevent HIV, and I know some of this work has been done here, and you're now doing prep work here that, if taken, is going to be very successful in preventing infection. There's only one vaccine study here on the list. This is the one from Thailand that was completed in 2009. That had partial efficacy, but all these other approaches are not vaccines or antibodies. They're mostly drug-based approaches. And so why do we need a vaccine? Why do we need a better way of preventing HIV? There's still more than 5,000 infections a day globally. I think some people, at least in the US, are losing track of that number. We've been so concerned about other emerging infections, and yet we're still having this many infections with HIV every day. And there's about 5 people infected for every 2 started on ARVs. And so it's not likely that we will be able to treat our way out of the epidemic. So we still are going to need other ways of preventing infection, we think, including a vaccine. And we know that one of the best ways to prevent a viral infection is to have neutralizing antibody. And so antibody is the mechanism we have for preventing virus infection, for preventing infection of the first cell by preventing attachment or by preventing entry. Once a cell is infected, our best mechanism for clearance will be T cells, and CD8 T cells in particular. And if HIV becomes latent, there's not good immune choices to take, and we'll talk a little bit about what could be done about that using some of the modern tools. So at the VRC, we're still working very hard to make better vaccines for HIV. That's still our primary mission. And most of the work now is focused on a trimer of the GP160 or a GP140 ectodomain of GP160, and having now the full crystal structure with all the loops and all the ways it connects to GP41, and now the full glycan structure on the trimer. We're working very hard to make a better vaccine to induce antibodies. So a lot of our program now is based around developing broadly neutralizing antibodies. And so we hope to be able to have a vaccine in the future. We're doing a lot of work on understanding lineages of antibodies and B cells that can get to make a broadly neutralizing antibody. But in the meantime, we're using the actual broadly neutralizing antibodies directly, either by delivering the protein or delivering it in a gene-based vector. So there's a long history of passive antibody used as serum therapy. In fact, the first Nobel Prize was given in 1901 to this man, Boehringer, for his work on making anti-serum in horses to prevent diphtheria. And he and Paul Ehrlich and others during that early pre-antibiotic era developed serum therapy for many infectious diseases, including tetanus and streptococcal infections. And these antibodies, mostly polyclonal antibodies, developed either in animals or in humans, were often used as a prelude to developing a vaccine. For all these different viral vaccines, the initial finding that antibodies were effective to prevent infection came from these studies and led to vaccine licensures in a few years later. So understanding how antibodies work and understanding how they neutralize the virus will both guide our vaccine development, but also, maybe be useful in and of themselves. I show this slide because I want to say a word about Butantan. Many of our big pharmaceutical companies started a little over 100 years ago, and many of them started on horse farms. And the reason it started on horse farms is this antiserum for diphtheria was usually done by immunizing horses and harvesting the horse serum, and that was the antitoxin for diphtheria that was so effective. And so this is a famous horse that was in the US that provided much of the antitoxin for children during those early days. But then it was also the horse that led to a lot of the regulatory process we now have for biologics, in the US at least, because after repeated immunizations, eventually this horse developed tetanus. And some of its antitoxin was contaminated with tetanus toxin, and some children died. And this is what led to the 1902 Biologics Act that established the Biologics Division of the FDA. It was the beginning of drug regulation. And so Butantan is famous for making antitoxins for snakes and poisonous frogs and spiders and other things by immunizing horses and using horse serum. And so Butantan also began a little over 100 years ago on a horse farm. Okay, there's many polyclonal antibodies that have been used against infectious disease. I'm just listing them here. I'm not going to go through all of them, but there's a long history of polyclonal antibody use for infectious disease. And there's now a lot of use of monoclonal antibodies in the clinic, but not that many for infectious diseases. Of the 30 licensed monoclonal antibodies in the US, only one, and now two, I guess, for anthrax are for an infectious disease. The other one is respiratory syncytial virus. Synagis has been licensed for almost 20 years to prevent RSV disease in high-risk infants. All these other, though, are in development in either phase 2 or phase 3 trials. So in the next coming years, there'll be many new antibodies, monoclonal antibodies, mostly human monoclonal antibodies used for infectious diseases. One of the big things that's changed for both vaccine design and for the development of new therapeutic antibodies is this technology. Starting with the ability to make monoclonal antibodies about 40 years ago, about 7 or 8 years ago, this technology to isolate single B cells, clone their immunoglobulin genes, and then reconstitute and synthetically produce monoclonal antibodies and discover new human monoclonal antibodies has revolutionized what we are doing now in terms of understanding the surface antigen structures of vaccine antigens and also discovering new antibodies for therapy. So now we're going to move to HIV, and because of these new technologies, many antibodies have been discovered that can broadly neutralize HIV by recognizing these different areas, different surfaces on the GP160 envelope protein. And in fact, as more antibodies are discovered, almost the entire surface of this pre-fusion trimer complex of the ectodomain of GP160 can be recognized by some antibody with broad neutralization. But most of these all have very unusual features, and one of the reasons it's been hard to develop a vaccine is because inducing these antibodies, either because of very long CDR3 loops or because of extensive somatic mutations, it's been very hard to induce these antibodies. We're hoping now that we have the full trimer structure, we'll be able to do that eventually. But in the meantime, we're going to use antibodies for either prevention or treatment. And the considerations here are quite different. So for prevention, which is what I'll mostly talk about today, what you need to do is prevent the infection of a single founder virus, which is usually what is infecting people. A single virus infects people. What you need in an antibody to prevent this one virus from infecting is very different than what you would need to treat someone who has a large viral load, either in their reservoir or in their blood. Putting an antibody into this environment with this large diversity of viruses will rapidly lead to escape. So in this setting, you need probably multiple antibodies, you need to maximize potency, and you have to avoid escape. Here, it has to be potent enough, but the biggest factor here is breadth of coverage. And because of PrEP and because of treatment as prevention campaigns, discordant couples are now off the table for prevention research. So it's going to get harder and harder to prove that things are working in prevention as we cross things off the list. So VRCL1 was one of the first, obviously, antibodies, human monoclonal antibodies discovered at the VRC by John Mascola. And it recognizes the CD4 binding site on GP160. And it has a lot of breadth and potency against viruses from all clades. It can neutralize viruses at less than 50 micrograms per mil. About 90% of those viruses from all clades can be neutralized. And so that was started in a program almost 6 years ago now to take into the clinic. It's been a long process, both to figure out the manufacturing approach, to go through the regulatory approach, and then finally to get into humans. But now we've completed a couple of phase 1 trials. We've completed a larger phase 1b trial with the HVTN in which multiple regimens and doses were evaluated. We've started some therapeutic trials, and now we've even opened a trial, an early phase trial in babies. And I'll tell you about the efficacy trial that also just started a couple of weeks ago. So these are the first 2 studies that were done, one in infected people, one in uninfected people. They were both dose escalation trials going from 1 to 40 milligrams per kilogram. In both studies, we compared 5 milligram per kilogram giving sub-Q versus IV. This is probably one of the higher doses you can give to an adult by sub-Q, and it's an important number to remember because I'll show you some data later that will make all of this sound more feasible. So what we found in the uninfected subjects is that these antibodies have good properties and behave as predicted. These curves were actually drawn prior to the study and are almost exactly what you see here, is that you can predict the peak antibody concentrations in subjects given a given dose. This is 5 milligrams IV, this is 5 milligrams sub-Q. The main difference here is that the peak antibody is a little attenuated in the sub-Q, but the trough level is almost identical to what you would have IV. And then you get higher levels given 20 milligrams or 40 milligrams per kilogram, and the levels surprisingly last above what we think could be a therapeutic level or an effective level for even up to 90 days. They also have virological activity, and so you can take a person who's actively viremic with millions of viruses, millions of genetically diverse viruses, and give them antibody and see a 1 to 2 drop in their viral, 1 to 2 log drop in their viral load that lasts about a little over 2 weeks before you start seeing rebound. This rebound occurs for a couple of different reasons. I'll just line everything up so you can see what it's like from the start, normalized to their baseline viral load. The virus escapes either because there's pre-existing resistant viruses in the pool, or because the antibody levels wane and the other viruses just start regrowing. And in a couple cases, there's 2 subjects who had resistant viruses prior to treatment. And so in these 2 subjects whose neutralizing activity against 10 selected clones of virus from subjects prior to treatment, their viruses were above 20 microgram per mil IC80s, and so these were the 2 who didn't respond well. So they had a lot of pre-existing resistant virus, so you wouldn't expect a single antibody to work in that setting. So again, even though in the beginning when they were infected with their single virus, this one antibody could have worked, once they have millions of viruses or billions of viruses and diversity, single antibody treatment won't work. So now some of these studies have been published, and as you see here, this antibody can sustain at relatively reasonable doses to deliver, 20 milligrams per kilogram, can sustain levels of neutralizing activity in serum for about 60 days. And so that gives you a potential for a 2-month regimen. Also, we know that from monkey studies, here being challenged with SHIV, BAL, that all the way down to 3 milligrams per kilogram can neutralize or prevent infection with some of these kinds of viruses. And if you look at the diversity of viruses, the number of the sensitivity of a big spectrum of viruses to VRC01, you see that this virus that I just showed you is on the easier to neutralize end of the spectrum. And there's viruses that go all the way up here, and other SHIVs, which are a little harder to neutralize on the higher end of the spectrum, can be neutralized by 20 micrograms per mil. And so if we can stay within this range, we'll neutralize many of the incoming founder viruses. And so the goal of this study, called the AMP study, the Antibody-Mediated Prevention Study, is to ask the question, can a passively infused monoclonal antibody prevent HIV-1 in high-risk adults? So using this VRC01, which is not our best antibody, but it has sufficient activity and breadth, we think, to answer this question. This trial has been started, like I said, a few weeks ago, and it's a placebo-controlled trial delivering VRC01 at 2 different dose levels, either 10 milligrams per kilogram every 2 months or 30 milligrams per kilogram every 2 months. It's randomized 2 to 1 with the treatment group as 2, and it's being conducted in MSMs and transgender people in North and South America and high-risk women in sub-Saharan Africa. It should give us power to detect a 60% efficacy and also give us a sense of what level of neutralizing activity in serum would be needed for protection against infection. So we are excited about this trial. We think it's a very important trial to set a bar not only for future monoclonal antibodies, but maybe for vaccines if we can make them in the future. So one of the things we've had to do is figure out how to make 60 kilograms of antibody in order to do this study. And so Dick Schwartz, I just wanted to show what has to be done. Not only do you have to show things work in animal studies and on a small scale in the lab, but you actually have to manufacture this at scale by good manufacturing approaches. And so Dick Schwartz is now making VRC01 in these 2,000-liter bioreactors and 2,000-liter batches. And to make the 60 kilograms needed for the AMP study, he's going to have to do 20 runs of this over the next 18 months or so in order to keep up with the needs of the trial. So, as I mentioned, it's not our best antibody. So one of the questions is, what would come next? What would we do in the meantime while we're trying to get an answer on whether a certain level of neutralizing activity would work? So the goals we have are to have broad coverage of at least 98 to 99% of all potential founder viruses or incoming viruses. We think that could take either 2 antibodies or maybe a bispecific antibody. We'd like this to be around 10-fold more potent than current antibodies so that we can give a much lower dose and maybe start getting it sub-Q instead of intravenously. We want to give that sub-Q injection every 3 or 4 months or maybe even every 6 months instead of every 2 months to either get it on a schedule, for instance, with Depo-Provera or other things and make it as convenient as possible for subjects. And we need to get it down, the cost down to what's comparable for PrEP or ARV drugs or to be added to these ARV drugs. And so where are we at with that? So this is a potency breadth curve, and I'm showing you all the new antibodies that are being discovered over time. And here's VRC01 up here. It has a lot of breadth. It's relatively potent, but not nearly as potent as some of these newer antibodies. But what you see is these newer antibodies that have different specificities, not to the CD4 binding site, they have less breadth. So they're very potent, but have less breadth. For instance, this CAP256 to the V2 site is a South African isolate that neutralizes almost all C-clade viruses, but it doesn't have as much breadth against other subtypes. But its potency is really extraordinary compared to VRC01. There's better CD4 binding site antibodies being discovered all the time. N6 is the latest one with even a much better potency than VRC01 and remarkable breadth. The antibody with the most breadth still is the one to the EMPER region, the membrane proximal region of the envelope, 10E8, which neutralizes virtually all viruses, but its potency is not quite as high as you'd like it to be. So we think that the best strategy is going to be to combine very broad antibodies with some very potent antibodies and have at least 2 in the cocktail. Another way to look at this is in these kind of potency graphs, these are antibodies along this bottom line. These are CD4 binding site antibodies, V2 antibodies, and EMPER antibodies, or these are to the glycan V3 and V2 regions. And so how far down, these are all separate individual, each dot is a different virus. So how far down you go is the potency, and how many are left at the top is the number, it indicates the breadth. So 13% are still resistant to VRC01, and MP and 10E8, for instance, only 2 are fully resistant to 10E8. And there's some extreme potency, especially in antibodies like this CAP256, that's also being now manufactured for development. So when you combine antibodies, you can see even an enhanced potency breadth. So now this is the breadth, this is the potency, and for instance, VRC07, which is a clonal, in the same clonal family as VRC01, is shown here with decent breadth, but and pretty good potency. But when you combine 2, which is on the brown curves, or combine 3 monoclonal antibodies on the gray curves, or combine 4 monoclonal antibodies on this black curve, you can achieve very high levels of breadth with very high levels of potency. So combining antibodies may end up being very useful. Another thing that can be useful is modifying the antibody or engineering the FC region here between the constant domain 3 and 2. Just a few amino acid changes can change their ability to bind the neonatal FC receptor, which is shown here. And I'm just showing MHC class 1 here because not because we're talking about CD8 T cells, but to me it was just so remarkable how much the neonatal FC receptor looks like an MHC class 1 molecule that carries the peptides for CD8 T cell recognition. So this slightly different type of molecule, this neonatal FC receptor, can bind this region of immunoglobulin FC, and it binds at low pH in the endosome and then allows it to be recycled back into the circulation, so you can maintain your levels of antibody much better if you have these what are called FC mutations. And MedImmune has a mutation called YTE that they use and have shown in clinical trials that this reasonably small dose, 3 milligrams per kilogram, given once to adults, can sustain titers above 10 micrograms per mil out to almost 6 months. So this is one example of a YTE mutation. We're using something called an LS mutation, and I'm just showing you some data here that's very new, and if you just look at, this is now a 5 milligram per kilogram sub-Q dose, and you give one dose and you can sustain levels for 3 months that are still up in the 80 or 90 microgram per mil range, and a dose after 3 months keeps it even higher. And so having antibodies behaving like this, maintaining such high levels or sustained levels over a long period of time, and now getting at relatively low doses, even for VRC01 and other antibodies, you could go down to 1 milligram per kilogram. And this starts making this look like it could be clinically feasible now to give a single dose that could be sustained. We're also considering the use of these to prevent mother-to-child transmission as an adjunct to the ARVs, which are good, but there's still about anywhere from a 1% to 3% breakthrough rate in mother-infant pairs treated with ARVs. And so using antibodies could potentially help protect against intrapartum exposure to HIV, but also could protect infants during breastfeeding period. And so if we can get these long half-life mutations, we think it's possible even to have a single dose of antibody in the delivery room that could protect the child through 6 to 12 months of breastfeeding. There's some evidence in animal models that this could work. This is work done by Nancy Hagewood in infant macaques that are challenged orally through milk exposure. And she's been able to show that if you can achieve levels of about titers of 1 to 300, or even sometimes titers of in the 80 to 100 range, that you can protect infants from these challenges through the oral route. She's recently shown, too, that even after infection, so you infect and then one day later start antibody treatment, and even though there's some evidence of local infection of local cells, the antibody treatment is able to clear and abort the infection before it's able to spread systemically, and you end up with antibodies even that are out here during necropsy. No virus could be found at all. So we think that these antibodies could have some role in maternal mother-to-child transmission prevention. So in summary, for PASI prevention, we have antibodies that can completely protect against infection in animal models. We're doing a proof of concept study now that would help us understand whether this could be a clinically viable approach. And we're working on improving potency by either engineering the antibody affinity or by combining antibodies, and we're working on half-life of antibodies that could make it more acceptable. How much time do I have? Okay, still a little time. So we're also using these antibodies for therapy. So in people who are already infected, it's not going to be as easy to use antibodies for therapy, but there may be ways in which they could be used that are being evaluated since we have the antibodies. One way would be to, if you could diagnose it early enough, give an antibody during acute HIV infection to diminish the peak viral load, and that should then diminish the reservoir and make it easier eventually to cure people. The other thing it could be used for is to give people drug holidays and maintain long-term viral suppression if we can find combinations of antibodies that could suppress virus off drugs during treatment interruption. And then there may be mechanisms or ways in which antibodies could clear the viral reservoir through Fc-mediated effector functions and bringing cells in to clear these virus-infected cells. Another technology that's being developed are bispecific antibodies. And most of these bifunctional approaches are trying to engage both an infected cell and a CD4 or CD8 or other type of cell with lytic capacity. And so if you can connect these two cells and get them closer together, despite the specificity of this cell, you can sometimes kill this virus-infected cell. So there's now bites and darts, and there's articles about those if you want to read more about it. They're just being used now in cancer treatment, and the first proof of concept in vitro is happening. We now have some evidence in vivo in monkeys that it can work, and these things will be moving along into manufacturing. So the lessons we've learned is that monoclonal antibodies are biologically active. A single antibody can suppress viremia by 1 to 2 logs. We've already learned that in treatment interruption with a single antibody, virus is easily able to escape. That's been shown now in 3 separate trials. And so a single antibody is not going to work for drug treatment holidays or treatment interruptions. We know that combinations of monoclonal antibodies will probably be necessary with high potency and high breadth for this type of therapeutic approach. And the impact on the viral reservoir will take long-term treatment, probably in combination with new ARVs and things that can activate cells out of latently infected cells. So in summary, we've had a lot of new human antibodies that continue to guide our vaccine development work and give us new therapeutic options. We have this efficacy trial started now for a neutralizing antibody called the AMP trial. Hopefully by 2018, we'll know if that is working. For treatment, we think antibodies can contribute to long-term viral suppression and potentially facilitate clearance, but this is somewhat still theoretical. We're improving the potency and durability of antibodies, and that can be an engineering project. And we know both from HIV and now from other virus endeavors with respiratory syncytial virus or influenza or coronaviruses that these antibodies can guide our ability to define structures and mechanisms of neutralization that can lead to better vaccine designs. So I'd like to thank all these people, especially John Moscola, who leads the VRC and directs much of the work that I just presented on antibody discovery and development, and also Julie Ledgerwood, who's now head of the clinical trials program at the VRC, to my great delight, and she's doing a great job of that. And I'll thank you very much and take your questions about HIV if we have enough time, and if Esper lets me, I'll tell you a little bit more about the Zika vaccine program in a few remaining minutes. Thank you.